is supporting partially this symposia or mini symposia, which is it's a program by ISME that try to, through the different ambassador, try to strengthen and promote microbial ecology as easily as broadly as possible across the globe. There is the early career committee, which is supporting and promoting early career scientists through different networking events, and the climate change committee, which is very important. It tries to bring the global attention to microbiologists, microbial ecologists, and how they can help in addressing the climate change emergency. And the SecCode committee, which work on trying to find the solution for naming the uncultivated bacteria and archaea that are identified through metagenomics, transcriptomic, and they are not culturable. With that, I would like to thank you, and please visit the ISME website. And if you have any question relating to our session, or to if there's something that I can help with relating to being a member in ISME, then please use my email and contact me. And right now I will introduce Dr. Amin, so he can give us a very nice talk on coral reef. Just a minute. Okay. So Dr. Amin Mohamed is I have a PhD in marine biology from James Cook University, and in Australia. After he finished his PhD, he had a postdoctoral fellowship in the marine genomics at Australia's Commonwealth Scientific and Industrial Research Organization. After that, he had different positions in Keele University and in the University of Queensland as a research scientist. And right now he joined the Marine Microbiome Myomics Lab at the New York University Abu Dhabi at NYU. And he's mainly interested in understanding coral adaptation to climate change by integrating state-of-the-art tools in genomics and microbiomics. And Dr. Amin, the floor is all yours. We are looking forward to hear from you
Thank you.
your talk. Yes.
Thank you.
You're welcome.
Let me share my screen.
Yes.
Just give me a minute. Can you see my screen now?
Yes, it's very clear. Yes.
Okay, cool. Thank you so much, Rahab and Sharif, for this kind invitation to talk to people in Egypt. I feel it's really important to keep connected with young scientists and new upcoming scientists and students, to share with them our experience and what we can do and how we can help. So today I will be talking about the coral microbiome and how we think that using the coral microbiome we can actually help corals with the increasing sea surface temperature and the increasing due to climate change. So I will give you a quick background. I know Rahab mentioned briefly, but initially I got my bachelor and my master's from Egypt, from Banha University. So this picture you see here is a reef flat of coral reefs and coral reef fish in Sharm el-Sheikh. This photo we took 15 years ago using my supervisor's camera. So my supervisor was Dr. Hani Abdesalam. He was one of the students of Dr. Hamid Gohar, a very well-known marine biologist back then in the 60s. And we were looking into these corals and trying to understand the ecology of coral disease. And we provided baseline information about what type of coral diseases are present in Egypt and how they are distributed across the Red Sea. And then I thought that this is not really enough to understand these unique ecosystems. You need to not just go diving and snorkeling and look at these corals, or even bring them into the lab to look under the microscope. You really need to look so deeper into their genomes to try to understand their functioning. So I went to this university called James Cook University in Australia, in North Queensland. Actually, it's James Cook is a mecca. We call it mecca of coral reef research, because I think it has been estimated that third of the coral reef research output was coming from this university. Not only because they are present in the center of the Great Barrier Reef, the largest coral reef on Earth, but also because of they started very early studying these coral ecosystems. So I was interested in studying the relationship between corals and the algae using genomics. I will talk more in detail in the talk about this. And then I had multiple postdoc fellowship, and then multiple research positions, until I started to work on corals again at NYU a few years ago. So talking about corals, corals are very simple animals. They are belonging to phylum Cnidaria, and these tiny corals. So here you see the coral polyp. Actually, I got asked this question more frequently. Are corals animals or plants? So actually, corals are, of course, the coral itself. The coral is an animal. Here is the coral polyp. And these polyps can build huge colonies, as you see here on the right, or down here on the left, or down here on the right. These coral polyps can build huge reefs that you can actually see from space. This is living organisms that you can actually see from space. How corals are able to build these huge reefs, so they can deposit calcium carbonate skeletons internal skeletons, by teaming up with algae of the family Symbiodinaceae, or we used to call them zooks. These are photosynthetic dinoflagellates that provide corals with the majority of their energy. Like, it has been estimated to be 90% of their energy needs are coming from these dinoflagellates. Not only dinoflagellates, but also corals team up with a diverse set of bacteria. We call it the bacterial microbiome, that are also providing very important functions and like nutrient cycling and vitamins to the coral. As you know, I think this simple principle that rules every single organism on Earth is no single organism live in isolation. So every organism is living in symbiosis with other organisms. So even a tiny diatom like this in the ocean, they are depending on many types or certain types of bacteria for their growth. So they attract specific bacteria from the ocean into the surrounding areas that we call it phycosphere, because these bacteria are providing vitamins and certain nutrients for the growth and development of the diatom. And this also goes to the human. So as you may know, we are more bacteria than we are human. So our body has 10 more times bacterial cells than our own human cells. So it's really important to understand that the organism now is called the holobiont. So when you study human or any kind of organism, you need to take into account their associated microbes. So back then, 10 years ago or 20 years ago, we dealt with microbes or bacteria as contamination. We need to get rid of most of them to study our own organism. But nowadays, actually, it's a central part of the organism that we need to consider when we do any experiment to understand the biology of any organism. So as any organism, corals also. So here is a coral colony of genus Acropora, and here is a zoom in into one of these tiny polyps. So actually, this is a coral animal. This tiny sac here with tentacles this is the coral animal. And this coral, here is a zoom in into the coral polyp. These corals, as I said, are associated with diverse set of microbes that includes dinoflagellates of the family Symbiodinaceae. These are intracellular micro eukaryotes living deep inside the coral tissue, providing the corals with 90% of their energy needs, and also a diverse set of bacteria that lives either on the surface mucus layer or in the tissues or even in the skeleton. One group of bacteria of the genus Endozycomonas are commonly known and usually studied in coral microbiomes because they show up in every coral, regardless of species, regardless of geographic location. So we thought that Endozycomonas is a core member of the coral microbiome. But unfortunately, we actually don't know much about their functional rules inside the microbiome. And also, regardless of Symbiodinaceae and bacteria, there are other types of microorganisms that we call cryptic that we don't know actually their function. So these include viruses, archaea, fungi, and even epicomplexans. So here also is another depiction of the coral polyp and different regions where you can find bacteria. So you can find it in different parts, and these bacteria are adapted to different parts of the coral to give specific functions. So here are a summary of recently published by what functions this coral microbiome can bring into the coral, and it revolves around provision of vitamins and also antimicrobial peptides to get rid of pathogens. But also, I'd like to highlight here, all these information were based on genomic prediction, which is actually you sequence, actually, sometimes you don't even sequence the genomes. Sometimes you get it from the 16S. So you infer functions based on the 16S data, which is really misleading. Even if you have the genomes, you actually don't know if, even if the bacteria have these genomes, are these genes expressed? Are these functions, are these proteins active? And there are many steps that can go wrong after gene expression. So post-transcriptional, post-translational, all of this also has to be right to get the function you need. But unfortunately, right now, most of the functions are derived from either 16S or genomic predictions. We're trying to change this with what we do. So the coral microbiome is, as I said, is a central part of the coral organism, and it's central to its health, and it's dynamic. When the coral is exposed to heat stress or any environmental stress, there will be some disturbance to the microbiome, but they can get the microbiome back and recover. So it's highly dynamic. So during healthy conditions, you have the normal microbiome here, and then when there's any disturbance, the fitness or the performance of the coral holobiont, meaning the coral, algal symbionts, and bacteria and all microorganisms, the performance will go down until this condition is recovered. So with this, you have a dynamic range of interactions. So mutualists or the beneficial bacteria will go down with the stress, and also this actually will be in the expenses of emerging of pathogenic bacteria and opportunistic bacteria that will lead depletion of these mutualists into the coral microbiome. This will lead to multiple things. First, it can lead to coral disease. As you see here, multiple coral disease, and they are visually classified. Like, for example, this one in panel A is called black band disease because it has here you can find a black band, and here is a white syndrome, which is recognized by this white area that actually kills the coral colony in this direction and leave behind the tissue here. So this coral white syndrome is a really important with frequently more recently we had so here you have a history of the thermal stress from early 90s until 2024. And as you can see here, these lines are indicating mass coral bleaching that was because these marine heat waves that we saw frequently in the previous 4 years. And this will actually affect corals very badly because this will disrupt the symbiotic relationship between corals and the photosynthetic algae, and this will cause bleaching. So here is a underwater photograph for bleached Acropora colony. And as you can see here is a picture from the Coral Reef ARC Center of Excellence in JCU. And as you can see here, you can actually see these coral bleaching from space. Here is a photo taken from helicopter. And then these white colonies you can see actually from space. This is very devastating to the coral because if corals don't get recovered after 2 weeks, they will be actually killed. This is just because absence of an important member of the microbiome, which is the Symbiodinaceae, and also changing the normal microbiome of corals. So what we are trying to do is we're trying to understand the response of coral responses to climate change by studying coral populations found in locations where there are extreme environments. So here, corals in the Arabian Gulf are exposed to end-of-century levels of sea surface temperatures. So by studying these corals, we can actually try to understand what mechanisms they have to try to combat these extreme environments. So corals in the Arabian Gulf are exposed to very high sea surface temperatures and also salinity. Temperatures can go up, actually it can exceed 36 degrees, which is lethal to corals. So to give you an idea, corals in the Great Barrier Reef in Australia will get stressed and bleached if the temperature goes beyond 31 degrees, which is 1.5 more than their maximum summer temperature. So we have a very good setup to study corals in this specific region. Hopefully we can, by learning lessons from them, we can help corals elsewhere. So what we do, we explore these corals using multi-omics. So we had a set of samples collected here from the region. We have this coral called Acropora pharaonis. We collected some healthy colonies and also some diseased colonies that have this white syndrome that's depicted here. So we have this white lesion, LS is a lesion, and this will kill the whole coral nubbin. And here is the adjacent lesion. So it's an area of the coral that's not yet diseased, not yet sick, but it's on the way of getting sick. And also DD, which is a dead colony. We also sampled a totally healthy colony that didn't get any disease, and also a healthy part of the colony that's already got disease. These samples were collected by scuba, washed on the boat, and then preserved in liquid nitrogen to obtain different omics data types. So these sampling events ranged from different temperatures that started with early signs of heat stress at 32 degrees and then high stress at 34 degrees. And during these two high temperatures, corals developed this white syndrome. And then we continued sampling at 30 degrees, 27 degrees, down to 25 degrees. We collected different omics, 16S ribosomal RNA umbilicons, to study the diversity of the microbiome, metagenomes, to study the functional role of these microbial assemblages, and also the metabolomes to study the whole holobiont metabolism. So this was using different Illumina techniques, and the metabolomics was untargeted metabolomics using LC-MS-MS. So here again is a colony of the infected coral colony. And as you can see here, the white region is the lesion. And we collected from this region, which is the lesion. Here is the adjacent to the lesion, and here is the dead. And here is a HD, which is healthy sample on a diseased colony. This is really important to understand the results that I will be discussing in the next few slides. So we start with all healthy samples collected at different temperatures, and we start to look at the beta diversity of the microbiome. And as you can see here, all these changes, of course, there are some changes, but they are not significant. So this means that the coral microbiome is really stable in this set of corals, despite being exposed to high degrees of temperature like 32 and 34. This is really amazing. Usually, as I said, corals in other regions will start to bleach even when they start 30 and 31. When we look at the actual bacterial families, the top bacterial families in these corals, with no surprise, we found Endozoicomonaceae. So this is a bacterial family that includes the genus Endozoicomonas that we saw. It's a core microbiome member of the microbiome, and also these not assigned or non-assigned bacteria. So this tells me that these corals harbor a unique set of bacteria of high thermal adapted Endozoicomonas, and also yet to be discovered microbes or bacteria specifically. This is all in healthy corals. When we go check the disease samples, we see a major shift, very significant microbiome changes that split these LS or lesion and the dead parts away from the healthy H and HD ones, where AL, which is an intermediate, actually having an intermediate position between the two groups. So what's happening in the actual changes in the bacterial families? You see H and HD are almost the same, dominated by novel bacteria and Endozoicomonas. And once you go closer to the lesion or to the disease, you start to see some changes that were very significant and obvious in lesion and the dead parts. And these significant changes were mainly coming from these Rhodobacteriaceae in green, and in pink is Saprosperaceae. These two bacterial families were peaked up, reached maximum in the lesion and the dead parts. So we think these are potentially pathogenic, potentially opportunistic bacteria that takes away the beneficial bacteria from the microbiome. When we do the differential abundance test, comparing adjacent lesion to the healthy, we found a few genera, this is genus level, a few genera that were highly enriched in lesion, in adjacent lesion, and mainly the top one was genus Orspera. So Orspera is a genus of bacteria that belongs to Saprosperaceae. It was 8-fold increased in abundance in AL compared to healthy. When we compare actually lesion samples, we still also found Orspera was the top that exceeds even 10-fold increase in lesion compared to healthy. So along with Orspera, there are other bacteria that includes alpha protobacteria and the other bacterial classes, but mainly Orspera was very obvious. So when we looked at the literature, very recently we found this interesting paper that describe a mechanism of predation, bacterial predation, by describing Orspera. Here, Orspera is this gliding bacteria here, and this is actually doing predation on a Vibrio bacteria. So they use hooks, grappling hooks to attach to the flagella of the bacterium, and then they puncture the cell to deliver toxins to lyse this bacteria. So this is a really interesting mechanism, and we are actually currently testing this in our samples. We're running an experiment as we speak to try to establish a relationship between Orspera predation on Endozoicomonas, because correlation between increase of Orspera and depletion of Endozoicomonas was very obvious, especially in the lesion. So it might be that Orspera is getting rid of Endozoicomonas, and then the coral is getting stressed because Endozoicomonas is providing something very crucial to the host. But what is it? What is important functions that Endozoicomonas is providing to the coral? So once it's not there, you can develop disease. This is what drives us next to understand the function of these bacteria. So in our set of samples, we have samples collected at 32 and 34s, and some of them are, remember, they are also still healthy. They are totally free from disease. So what's really unique about these corals that protect them and not get them having any kind of disease? So we started to look at our metabolomes. And then as you see here, the metabolome really nicely clustered well with the samples. Here you have healthy and the HD samples. In between, you have the AL samples. And then in red, you have the lesion samples, and the dead samples are clustering far away. So this is really nice. So you have a clear response from the metabolome that reflects the disease condition. Okay, so what are actually these metabolites that are driving this transition between the different healthy conditions? So we identified 279 predicted metabolites or annotated metabolites that we can use annotation from. So we have 8,000 metabolites, but out of these, we have a few hundreds that have annotations. And out of these, we identified these 279 metabolites. And then we checked their presence and absence in the different conditions, healthy conditions, and also diseased conditions. Here is an upset plot. It's like a Venn diagram to show the interaction between different metabolites in the different conditions. So we think that these metabolites are really important for the healthy conditions and also the disease samples. So we used this small molecule pathway database to check these metabolites should be actually targeting specific pathways, really important for health and disease, not just randomly hitting any pathway. When we check this, we actually found that there are significant enrichment in specific pathways, as I mentioned, and the top 5 ones were related to steroids, and also to polyamines. So steroids are including mainly steroid hormones that are very important in vertebrates and eukaryotes in general. And also polyamines, spermidine and spermine are these polyamines that are very important also in regulating growth and the important functions of the cell. So, okay, this is really interesting and cool, but who is making these compounds? Who is making these molecules? So we started to look at metabolite co-abundance networks. So here we have a network, and each node is a metabolite, and each line is each edge is a connection. So either positively connected or negatively connected, positively in red and negatively in blue. And all these metabolites can have different functions into the network. So some are present in the periphery, some present in the central, some have very small number of connections, some have high number of connections. So by doing these networks, you can actually pinpoint to specific nodes that we call it hub nodes. Hub nodes, that means if you lose this node, the whole structure of the network will be distorted. By applying this principle into this network collected using metabolite abundance in all healthy conditions, healthy corals, we can also do the same and study what happens to the network when you go to the adjacent lesion and to the lesion. And here I'm highlighting here one key node that has the highest betweenness centrality, which is one of the important measures that can define these hub nodes. And as you can see, this one is petrosine. Petrosine is a precursor of all these polyamines. As you can see here, petrosine has a central position with high connectivity in healthy, and this gets reduced when you go to the adjacent lesion, and this get largely reduced also in the lesion. So we think that these network metabolite hubs with the highest betweenness are very important to this transition from healthy to lesions. And these polyamines are so important in this transition because they are known for growth regulators and actually heat stress tolerance. Okay, that's interesting, but this is all based on correlation. So what? Okay, this goes up, this goes down. We need actually a more valid proof of what's happening. So we looked more deeper into the subnetwork of this subnetwork that has Petrosine. So what is connected to Petrosine? And then we found the highest connected ones, positively connected ones, are actually steroids. That includes steroid hormones. And of course, this, when you go to the lesion, this got depleted. So there is no more connections to these steroids. Okay, but now we have also our metagenomes, and we can mine these genomes to try to understand where these functions are coming from. So we assembled the holobiont metagenomes. So the metagenome will be coming from corals, from algal symbionts, and from microbes. But as you see, the majority of the majority of the metagenomes are coming from the coral and the algal symbionts, and a tiny fraction only coming from the microbes. We assembled these reads into contigs, and we used KIG database to find the pathways. This is using the metagenome, and then we try to ask if these genes are shared among all holobiont members, or they are unique to one specific member, or here, for example, microbes, or coral, or algal symbionts, or shared. And we did the same also for the metabolomes. We have these metabolites. We can connect to their compound IDs and also getting their chemical reactions from KIG. And these chemical reactions are actually happening because of these enzymes. So using also the metagenomes, or the genes that we predicted, we can actually do the same. And of course, as you expect, there are many of these functions are shared across all these members. But I'd like to draw your attention to these two specific pathways: steroid degradation and steroid hormone biosynthesis. Steroid hormone biosynthesis was mainly dominated by the host, while here steroid degradation is mainly dominated by microbes. Okay, so here we have a clear pattern that microbes might be involved in specific pathways in the holobiont. But who actually is making these or contributing with these functions in steroid degradation? We didn't know. So to do this, you have to assemble your metagenomes into what's called MAGs, or metagenome assembled genomes, where you can assign function to specific genome with a taxonomic label. So you can here is like a few depiction of how would you assemble these MAGs. So you extract your DNA from the community, you sequence it, and then you do the assembly. You have the assembled contigs, and then you do binning. You bin them based on different like abundance and different chimers. And then after binning, you do dereplication to get rid of non-redundant genomes. And then at the end, you have a set of non-redundant MAGs or genomes, and then you can map out the abundance of these MAGs in your metagenomes. So we obtained a few bins. Among them, we only found 11 MAGs that were dereplicated by DREP and all high quality. And one of these MAGs were actually one for endozoicomonas that was very high quality according to ChICAM. So when we go back and check which MAGs have these genomes, was a big surprise that we found that endozoicomonas MAG was the only one that has these steroid hormone degradation genes. So this was very fascinating to see that endozoicomonas has this rule that's unique to the whole holobiont, with providing the holobiont with genes that are involved in steroid degradation. So to confirm this, is this happening only in corals here, or it's all over globally? So we obtained all endozoicomonas genomes from different species, from different invertebrates, and also a free living isolate. And here, what you can see here is a band genome. So each track is a genome. And when you have a dark line, it's a gene cluster, presence of a gene cluster. If it's fainted, so this means the gene is absent. So the band genome actually consists of three genomes. One is the core genome, where you can find this set of genes in all of the genomes, and also some accessory genes or genomes, where you have this set of genes present in some and absent in other genomes. And also you have singletons, where you have only these clusters present only in one genome. Okay, so now we can ask many questions about this. We also predicted biosynthetic gene clusters, but I will not bore you with all the details. Also, we have here heat map that shows the ANI's percent similarity to see that MAG10, here is our assembled MAG for Endozoicomonas, cluster very nicely with Endozoicomonas acroberi, which is Endozoicomonas actually isolated from acropora. When we looked at this key enzyme KSTD, which is involved in steroid degradation, we actually can see that this gene is common more or less in at least 70% of the Endozoicomonas genome. So it's actually a very common function in Endozoicomonas, not just in our locally assembled MAG. This work actually still under review in Nature Communications, so hopefully it will go online soon. Okay, so now we have our metabolomic data, and we can now check the abundance of these metabolites across different conditions. And so here you can see the abundance of these steroid hormones in the host. And as you can see here, the majority of these metabolites are going down with temperature and disease in every case, except these three terminal steroids: progesterone, estradiol, and testosterone. They actually have an opposite trend. They go up at 34 degrees. So this was really interesting. So how come corals can produce these terminal steroids while the precursor, which is cholesterol, is actually depleted in 34 degrees? So this was like a question that how this is happening, and we need to dig more into this. And we also check on the steroid degradation, and we found these genes that can take testosterone and degrade it into androstenedione down all down to HIP into the TCE cycle. So actually using testosterone as a carbon source. But given that Endozoicomonas is able to do this conversion from testosterone and androstenedione, and actually many bacteria can do this. It's not, but not coral-associated bacteria. So other known bacteria can do this transformation to make use of steroids as a testosterone as a carbon source. But actually, it can do the opposite way, because these enzymes usually works both ways. They are reversible. Okay, so to confirm this, that actually Endozoicomonas can actually provide these terminal steroids to the coral, given it has this unique ability to degrade testosterone, we actually grow Endozoicomonas in culture. And then we did this experiment where we grow Endozoicomonas in the lab with cholesterol and normal temperature, and then 34 degrees at high temperature. And what we can see here, after growing, after incubating these Endozoicomonas with cholesterol, we actually can map out a pathway of actually biosynthesis of these steroids from cholesterol down to testosterone and also down to progesterone. So we can see this with statistical significance. So actually, yeah, all these molecules are produced when they are exposed or incubated with cholesterol. We also confirmed this annotation for progesterone and testosterone using mass spec. So we are pretty sure that these compounds are what we are talking about. So this is really interesting because these terminal steroids are actually eukaryotic signals. And bacteria are not supposed to produce these testosterone or progesterone. And actually, there are some bacteria that can produce cholesterol, but there are a few very select bacteria that we know that can do this type of transition. So maybe this function unique to Endozoicomonas is one of the key functions that makes it very that makes coral are very relying on them for their functions, because these progesterone and testosterone also have these functions for priming innate immunity, so they can actually defend off pathogens. And this what actually fits well with what we see in the microbiome. So to sum up, so corals in the region, in the Arabian Gulf, are highly stable compared to other regions where the microbiome are shifting with the seasons. Here they are very stable, dominated by a select group of bacteria, including Endoxycomonas and some novel bacteria. And actually, I hope I can, I was able to convince you that multi-omics actually can provide some novel insights into these symbiotic interactions. And what we show is the Endoxycomonas can actually manipulate the host steroids, mainly cholesterol, to make these terminal steroids as progesterone and testosterone that has no functions, known functions in bacteria and are mainly eukaryotic signals. And these steroid hormones can potentially prime the host innate immunity and actually reduce inflammation during thermal stress. With this, I'd like to thank all members of the Marine Microbiomics Lab, Shadi, Michael, and especially the coral team, Yanya, Sebastiano, and Amitash, and also the Marine Biology Lab with John Burt and Trent, with whom we collaborate a lot in multiple projects. Thank you so much for your attention.
Thank you so much, Dr. Amin, for the very insightful presentation. Do we have any questions from the audience? You can ask your questions on the Q&A session or in the chat if you would like. Do we have any question for Dr. Amin?
I would like to ask a question.
Yes, definitely.
First of all, it's a really fascinating talk. Once again, from someone who studied infection biology in human, I think this is really cool. That you are able to take a study from the macroverse into the very, very detailed molecular details of the study. It might be a very naive question. So do you think, for example, adding Endoxycomonas, for example, for the dead corals would help them to get revived again or something? Or
I would say no. For dead, actually, once
Mm.
you reach to the death,
Mhm.
I think there is no return. But
Or the
with
infected ones?
the infected ones, I should say theoretically yes, but we
Mhm.
are testing a couple of these. So this is coming from the idea of probiotics, and
Yeah,
actually there
exactly. Yeah.
are some people actually working on this, and including us. We are looking for these certain bacteria and actually cocktails of bacteria to use to help corals, reduce the stress
Yeah.
during thermal stress.
I understand this point. So actually, so the other question is basically there's like a fine tuning or a thin line between adding Endoxycomonas, for example, or such probiotic bacteria that can lead to, for example, the excess of probiotic bacteria can lead to kind of like biological imbalance that can cause infection into the corals, because this is quite common, for example, in humans. Like there is the beneficial bacteria, but if you add more of them, it can cause a certain biological imbalance. So is it the same for corals?
No, we haven't tested this, but this is actually fascinating. So if you have more of these beneficial bacteria,
Yeah.
it can have a detrimental effect.
Some of them, yeah.
Okay.
Some of them, if you have, for example, biological imbalance, for example, if you are immune suppressed and you give a certain
Yeah.
beneficial bacteria, it can cause a certain infection.
Actually, I think it can happen because Endoxycomonas actually belongs to gamma protobacteria, where Vibrio actually most of the
Mm-hmm.
like known pathogens in humans and corals as well belong to. So potentially, yes, I think so.
Okay. Yeah.
So I think it has to be a balance, as you said. So all these has to be present in specific abundance, certain abundance. And once you have a disturbance, things go out of control. And then you have, other bacteria. So Orspera, for example, is not part of the coral microbiome. It's introduced and it's opportunistic bacteria. So when we check the healthy corals and we check the abundance of these Orspera, they are not nothing.
Okay. Yeah, that's
Yeah.
very interesting, and it's great that actually that your metabolomics are quite aligned with your genomics. That's quite very rare as well.
It's usually rare, I know. Yeah.
Yeah.
I've been in multi-omics for 10 years now, and usually if you do, for example, DNA methylation and gene expression, they are no correlation or at all, or
Yeah.
very small correlation, but luckily we have some good agreement between them.
Yeah, great talk. Thanks a
Thank
lot.
you.
So we have a question from the audience, from May. She is asking if coral microbiome become more resilient after recovering from bleaching event, which is actually it's a interesting question for me too. So if they recovered from a bleaching event, would they be more resilient or not?
I think this hasn't been tested before, but I would assume yes. So this is getting when you get exposed to stress second time, you have a history of the exposure, so you have an experience with this. And actually this is related to epigenomics, because when you exposed to certain activity and then you learn from it, what set of genes should be expressed or not. So second time, when you, as immunity, you get immune to it.
Yeah.
So I would assume yes. We have a small data set that we are working on right now with people in Egypt. Actually, a very nice group of actually very small group of people, Hadir and Dr. Mohamed Yusuf from Bursaid University and Swiss Canal University, and we have the same set of data. We have corals before bleaching, during bleaching, and after bleaching, where they actually recover. And these samples are in the sequencer now as we speak. So hopefully by next week or so, we will have the sequence and we can reply to your questions, May.
Soon.
Thank you so much.
Do we have any other question? Okay, maybe if there anyone who have other question, it could be asked during the roundtable discussion. Right now, I would like to introduce our second session speaker, Dr. Raed Abed. Dr. Raed is a full professor and the head of the